Hey, how many of you got to see Blake's lecture yesterday? Wasn't that absolutely amazing? Very, very cool. CRISPR, but not the classic CRISPR-Cas9 genome editing, more of the way that CRISPRs work in terms of E. coli and other bacteria, and the way all of the different protein components interact with each other. He did give a similar seminar for the NIH Institute of General Medical Sciences probably about 6 months ago, and I think that's online. I'll see if I can find the link for that. A lot of very similar things, some amazing movies about how all of the different molecular machines fit together to do this really amazing bacterial immunity system. So that being said, we can move forward with today, which is more and more of this stuff, really all about virion entry, which of course is the release of the genome into the cytoplasm, and in many, many cases that also needs to get to the nucleus. So we talked a little bit about some of the receptors last time. We'll talk more about some of those today, and we'll talk more about the individual viruses once we get to each of those. One of the really critical aspects, particularly of animal viruses, is this membrane fusion concept. So you've got the envelope around the outside of your virion. The genome, of course, is on the inside. How do you get that genome on the inside out to where it needs to be? You have to have membrane fusion that takes place. Yeah, Ian?
For the hemagglutinin fusion protein?
Yeah.
I'm sorry, last time you said it was uncoated, or it was revealed in the hidden protein by the degradation of the protein covering it under acidic conditions?
Okay, well, we'll get to that a little bit later on, but it really has to do with the fusion, the exposure of the fusion protein, and we'll look at that a bunch today in different parts. In the case of influenza, which we will talk about a lot more when we get to talking about orthomyxoviruses later, yes, it is the hemagglutinin protein that gets cleaved by a specific protease, and that cleavage allows the fusion peptide to push itself into a membrane and pull those two membranes together. So it's about the membrane fusion process. And a lot of these things, transport is all a cellular mechanism. The viruses take advantage of all the cellular mechanisms. Also true for all the receptors. All the receptors, there's no specific virus is going to attack to this cell because it has a very virus-specific receptor. All these receptors are used for other things. And again, when I say receptor, I mean pretty much any kind of molecule on the cell surface. And then we'll talk a little bit about some antiviral drugs, because a lot of those are involved in trying to block these virus-cellular interactions. And I did finally get the videos to work on these things. So let's take a look at first the HIV fusion. Again, we'll talk a lot more about these individual proteins later on, but this is a really nice molecular animation of how, in fact, a very similar kind of fusion protein, in fact, to HA, undergoes this fusion process. So you have the viral membrane down here, the cellular membrane up here. This is just one of a trimeric protein, so 3 monomers come together. The really important thing right here is this hydrophobic part of your protein, which is sticking up right here. You notice this conformational change. This is the conformational change. Remember, viral membranes down here, cell membranes up here. It's pulling these two membranes together. And this is all based on various different both structural and biophysical measurements of interactions between HIV and cellular membranes. So you'll start to see the conformational change happening right here. The two membranes are being pulled together, and then finally you have membrane fusion that takes place here. You always have a hemifusion intermediate. That's one complete phospholipid on this side, and then these two, which are in the middle, the last step where you pull these two apart and you end up having this complete membrane fusion is a still pretty open question in terms of exactly how that happens. But once you've gotten these two membranes apart, the virus nucleocapsid here can get through into the cellular cytoplasm in this particular case. So sort of review kinds of things here. It's really about that conformational change and getting the two really hydrophobic parts of the protein pulled together. And so this now transitions into some drug design processes. Understanding this particular mechanism allows us and other people, of course, to design drugs that will now stop this conformational change from happening. And these are some of these drugs that are doing that particular process. There's one small inhibitor here. I'll finish right at the end. Interacts with the helix, which you notice is moving around and blocking it from undergoing this conformational change to pull the two membranes together. I don't think the molecule actually is a drug yet, but it's one of the kinds of ideas that are trying to use now. Understanding the molecular details of this process, and then being able to design or essentially find a molecule that really fits perfectly into one of the stages of the velocity for the conformational change. Yeah. Pro 140. Yeah, so I don't know details on that particular. There are surprisingly very large number of anti-HIV drugs that are out there. This is one example of those. I think that one is actually an antibody-based process. I'm not absolutely certain. But if you send me an email, I'll try and get more details on it. I'll let you know more about that. So that was the HIV fusion process. Again, understood very well. This is a similar kind of process. Now looking at we can go back to here. We're going to now look at dengue and how the dengue virus actually uses to the now no longer external membrane. HIV actually fuses plasma membrane. What dengue does is once it's got inside the endosome. And in that process, there's actually a change very similar to what happened with influenza. In terms of HIV, we didn't talk about this later with HIV. It has to do with actually individual receptors causing this conformational change. But dengue has been a different process. So now we can take this dengue one. The beginning talks about how dengue is nasty diseases. But now we're looking at the dengue virion here. So comes in through receptor-mediated endocytosis. Not quite sure why it's going sound here. Should we turn it off? There we go. So here in the endosome, pH change, big conformational change of this surface protein. And Dr. Alec Hirsch, who's giving the guest lecture on these kinds of flaviviruses, will talk about this process. Again, a hydrophobic piece here, a hydrophobic piece here. Those then get pulled together in a conformational change. And you'll notice all the conformational changes happening in these viral glycoproteins, pulling these two membranes together. You get a hemifusion intermediate, then those two pull apart, and then the genome segments, which are on the inside, you can just barely see them here, end up being released. This is a negative strand RNA virus. Actually, no, sorry, positive strand RNA virus. get that one wrong. -1 point. So that fusion process, but again, it's the idea of the enveloped virus, two hydrophobic parts of your receptor protein, which then allow the two membranes to pull together and release the genome. In the case of dengue, in the case of HIV, it's actually a nucleocapsid, so it's the genome cased in some extra protein above and beyond that. And no, we're not interested in this, thank you. We can now close this guy. We'll get back to this other one later on. Yeah, do you have a question?
So that was the uncoding of the...
Yeah, so I would say this is really the membrane fusion process which is taking place. We'll talk about uncoding. Uncoding usually has to do with removal of proteins that are associated with the genome. And in the case of dengue, the genome is released then directly. So it's the fusion and uncoding which are simultaneous. In the case of HIV, where you have a separate nucleocapsid, which is true for quite a few of the enveloped viruses, you have to have that extra step of the nucleocapsid breaking down before you can get to the genome. So there are multiple steps there. Yeah, Al?
In the case of dengue, is the virus actively initiating endocytosis, or is it just being passively taken up and then...
Okay, so the question here is basically what's that actual entry process, getting that genome in. In the case of dengue, it is a receptor-mediated endocytosis, so there is that interaction of the receptor, and once there's an interaction with the receptor, then endocytosis is stimulated. So it's not doesn't seem to be a just random, as it were, process. It's definitely dependent on receptor virus glycoprotein interaction. Yeah?
So with the dengue, because it's a positive single-strand RNA, it doesn't need to get into the nucleus, so it just releases into the cytoplasm.
Yeah, so great comment, Angela. Basically, again, that's why I misspoke on the positive strand RNA viruses. But it's those, a positive strand RNA virus, and any of these RNA viruses, they don't necessarily have to get to the nucleus in order to be able to replicate, and so they'll just in the cytoplasm. So that released genome is at that point active. And so it can go ahead and be translated to the positive strand RNA virus or get replicated immediately at that point. You've got DNA viruses, and it turns out for some of the other RNA viruses that we'll talk about later, those end up replicating in the nucleus. But in this case, no, that genome is released immediately in the cytoplasm, so you're exactly correct. HIV is different, and we'll talk about that. HIV needs to get into the nucleus, having to do because it's going through that DNA intermediate, and anything that's got DNA associated with it in a eukaryotic cell, DNA in the cytoplasm does absolutely nothing. Actually, it induces antiviral responses. That has to get to the nucleus. So any kind of DNA virus that's interacting with the eukaryotic cell has to get to the nucleus. Some of the RNA viruses do. All of the retroviruses do because they've got that extra DNA step which is involved. Okay, we're all happy on fusion? Yes? When you ask a question like, oh, one more question before I ask you a question. So you can get your clickers out. Yeah?
So I was wondering if, since it's hanging out in the cytoplasm, are there any modifications done to allow it to not be degraded?
So the question here really has to do with, how does the virus deal with the fact that extra RNA hanging out in the cytoplasm is probably a bad thing? Well, there's certainly messenger RNA that's in the cytoplasm. And so if the viral RNA can pretend that it's messenger RNA, it's perfectly fine. The real problem is when you have double-stranded RNA. And we'll talk about that when we get to some of those replication intermediates a little later on. Okay, so clicker question. Fusion of animal virus envelopes with host membranes during infections seems to mostly happen by blank fusion proteins, proteolysis of, structural changes in, pH-induced aggregation of, nuclear localization of, endocytosis of. You guys better get 100% on this.
No pressure.
We had 2 last time. So, and I did upload the clicker scores. So if you're not seeing a clicker score associated with your login on D2L, let me know because I will try and figure that out. Send me your clicker number.
But I don't know if that's what's in the
Five. Okay, get me move these over so you can see. No 100% this time. But yes, it is definitely the structural changes that are happening. And that was sort of the point of all of these movies, with all of the structural changes happening in these. You can get the production of some of these fusion peptides due to proteolysis. But that's not giving you your fusion itself. pH-induced aggregation, that's what happens in the dengue. So you actually have a dimer, which we didn't really talk about, which goes to a trimer. And then also pH-induced changes like you have in influenza. But again, that's just making everything ready to have this fusion process take place. And then, nuclear localization. There actually are a couple of viruses that fuse at the nuclear membrane. And I needed something else, so endocytosis had to happen as well. So this is getting back to Angela's question about the nucleus. Why do you have to get to the nucleus, and why do so many of these guys over here have this nuclear localization that's part of this entry process? Again, clearly DNA genomes, everything's happening with DNA is happening in the nucleus. So anytime you've got a DNA, there's a double-stranded DNA virus, a single-stranded DNA virus, a retrovirus, all of those guys have to go to the nucleus in order to replicate. But it turns out that there are a few RNA viruses that do that as well, that actually have nothing to do with the reverse transcription. Because reverse transcription, again, that's always going to be a DNA-related thing. But if you think about it, select the answer here. That would actually be good, so people actually get points for it. That would be good. Those of you in the 77% that got it correct. So, yeah, all those extra points. But it turns out that, not surprisingly, RNA synthesis, if you think about where the messenger RNAs are actually made, where are they being made? Messenger RNAs are normally being made in the nucleus. So all of the precursors for RNA polymerases are going to be in the nucleus. So even if you do have a positive strand RNA virus that's being replicated in the cytoplasm, somehow it's got to get all of the ribonucleotide triphosphates, all of those things to the cytoplasm, which is usually not where they are. So in some cases, you're just thinking about it, where is all of the nucleotide polymerization happening in a eukaryotic cell? It's all happening in the nucleus. If you're a bag of nucleic acid, where do you need to go if you want to replicate your nucleic acid? Going to the nucleus. So there are lots of different processes for doing this. And a couple of those different ways are, first, wait till there's no nucleus anymore. That actually works reasonably well. So, mitosis, nucleus breaks down, then you can get yourself to the nucleus in that process. You don't have to worry about the whole nuclear pore complex, getting your genome in, getting your nucleocapsid in. Turns out that most retroviruses actually do this. They hang out in the cell, wait for it to replicate, and after that mitosis process, in that way, when the nucleus reforms, it reforms around the virus. And they seem to really work quite well that way. You also have examples of where viruses have taken advantage of the normal nuclear transport machinery and take the genome and move it into the nucleus, together with standard nuclear import proteins. You have a couple of cases where the capsid itself, either of a naked virus or the nucleocapsid that was present inside the envelope, that gets moved to the nuclear membrane and then gets dissociated at the nuclear membrane. So this is getting back to Ian's question about the uncoating. So you have the release of the genome from the proteins that are around it, just literally at the nuclear membrane, and that genome itself gets transported through the nuclear pores, and then in very few cases, and this is particularly true for a lot of the very small virions, the whole virion gets brought into the nucleus. And this is true for hepatitis B and SV40. But again, these are very small kinds of virions. Now, this is true for all the eukaryotic systems. What about archaea? We don't really know. We'll talk about that right at the end of the class. But bacteria are much better understood how this works. For bacteria, how many of you are taking micro right now? Okay, so lots of you. I'm going to give a guest lecture on viruses, so you can sort of sleep through that one. No, I will talk about different stuff in in the lecture there. But as all of you in microbiology, and hopefully those of you who are not taking microbiology know, bacteria have a real problem, and that is having to deal with the rest of the world. And the way they deal with that is multiple membranes and or cell walls on the outside of the particular bacterium, which is a problem if you think about this from a virus point of view. And so viruses have evolved these really amazing mechanisms for getting through those membranes. And probably the most amazing, as far as I'm concerned, is what goes on with T4. Again, my apologies if you've seen this video a gazillion times, but it bears watching again. It's really absolutely amazing. And extremely well based on the structures which are known here. So here's our bacteriophage T4 virion coming down and interacting with the poor unsuspecting E. coli's who are hanging out here in the background. So here comes the virion moving through. Now there will be, of course, a lot more of these virions here, very low MOI that we're talking about. The tail fiber proteins here are going to interact with receptors on the outer membrane of E. coli. E. coli is gram-negative, so it's got two membranes here. And it's two different receptors that they interact with. First those here, then these here. And then there's this compaction process. This is the drill, which literally drills the part of the tail through the first membrane. Then there are enzymes that degrade the inner membrane, and the genome is actually released. This is a, okay, we can don't chase the wind, even though I really like windsurfing. We're not going to do that one. This whole process of, again, it's still a conformational change, but now instead of a conformational change pulling the two membranes together, it's a conformational change which stuffs the part of your tail through the membranes in order to be able to release the genome. This movie was shown at a scientific conference I went to, actually probably about more than 10 years ago now. It's kind of frightening. But everyone was like, oh my God, this is just absolutely amazing. And then I talked to one of the guys who worked on all of these structures, and this is almost atomically correct, the whole process there of the video, with one real interesting exception. And any of you know what that is? Have I told you before? The compaction of the tail is actually incorrect. It should go the other way around. So again, not that it really matters, but so now I know what the problem is with that particular video. So that's pretty classic for bacteria, particularly the bacterial viruses that have what's called the contractile tail. And so these will start out with long tails. They compact the tail, again, conformational changes in the proteins, and that then literally pushes the genome transfer apparatus, this little tube actually that the genome comes through, and that then gets into the cytoplasm of the bacteria. And we'll talk about some of the other processes. In fact, towards the end of the lecture today, we'll talk about MS2, which actually has a totally different mechanism for getting inside the bacterial cell. So because of, let's get back to here. Because of the fact that this entry process is clearly something that's very important for getting your virus genome inside the host, If there was some way that you could block that, then you're not going to have virus infection. You're not going to be leading to virus disease. So we looked at that one particular case in HIV where it's blocking the conformational change, which is happening in order to get the two membranes to fuse with each other. Vaccines basically go one step before that and try and block the interaction of the virion with the cell in the first place. These are either antibodies or in terms of, how many of you had immunology? Know what an antibody is? Think you know what an antibody is? That would be me. I have never had immunology in my life, so that makes a lot of us. But the idea here is that the antibody is going to interact with the surface of the virion, and once it's interacted with the surface of the virion can't interact with the cells anymore. So that's, I think, the easiest way to think about what's going on here. So it turns out that a number of, and in fact, maybe even this therapy that we're talking about for HIV, people are now using antibodies as drugs to try and block some of these kinds of interactions. And another way that people have tried to do this, instead of looking at it from now what the virus is going to interact with the cell, well, if the virus is always going to interact with a specific receptor, what if you just flood the patient, wherever it is, with a bunch of receptor that's not associated with the cell? So as I told you for HIV, what happens when you have interaction with the receptor, you have this conformational change that happens, which releases the nucleocapsid. Well, if you could trick the HIV into fusing with something that's not a cell, the nucleocapsid is released extracellularly, it's not going to be able to do anything. So this was a really promising kind of therapy, ended up actually not working out terribly well, but people are still trying to do some of these things. That working with antibodies is absolutely fabulous in terms of vaccination because it's your own antibodies that are doing this. Making separate antibodies and trying to use those as drugs is a lot harder and very, very, very expensive. Probably a lot of you have heard about the really expensive therapeutics in lots of different ways. Many of those are antibodies. They end up being tens of thousands of dollars, if not hundreds of thousands of dollars per treatment. So it's a very, very expensive proposition. On the other hand, small molecules, like the one that we saw in the animation about HIV, these are much, much, much cheaper and actually easier to get into people for regulatory reasons, but also just for pharmacokinetic reasons. And so a number of these are being tried. Certainly the HIV one that we talked about. Influenza, there is what's called a M2 channel inhibitor. This has to do with the pH changes. If you block pH changes from happening, you're going to block conformational changes from happening. And that used to be one of the first-line drugs for influenza. It turns out the influenza is mutated and the drug doesn't work anymore. There also are a couple of other ones. The names of these are not critical. I had a guest lecturer once who was an MD in a previous life, then was a PhD student, now he's back to being an MD, gave this, and so he knows all the names of these things. I always forget the name, so I won't expect you to remember it. But they have to do with trying to use these small molecules to block uncoding, and really it's those conformational changes which are being blocked. So let's take a quick look at some of these. This is Amantadine, which blocks the ion channel in influenza virus, which is really important for that conformational change which happens. And it literally sits down right here in the middle of this otherwise pore. So the ions would normally go through here. This small molecule blocks that. Poliovirus, it's a little hard to see here, but this is one of those three. You remember VP1, VP2, and VP3. We'll talk a lot more about them later when we talk about poliovirus. This undergoes a conformational change in order to release the poliovirus genome. And again, one of these small molecules, a little bit easier to see, pardon me, on this side over here. In pink is sort of sitting into one of the three capsid proteins in poliovirus that prevents the conformational change that allows the genome release. And the last one of these that I wanted to talk about, anybody heard about Tamiflu? Probably did when people were worried about flu outbreaks. This is one of the first lines of antiviral defense mechanisms to be used. It blocks the other protein which is on the surface of flu viruses called the neuraminidase. That neuraminidase is very important actually for cutting off the receptor that you have on flu viruses and allowing the flu virus either get inside the cell or to not be stuck on the cell as it's being produced. And again, it sits down right in the middle of the active site of this enzyme and blocks the activity of that particular enzyme. So that's almost all that I have to do, I wanted to talk about in terms of entry. Any questions about this before I ask you a question? Okay, I can ask you one. Good, we'll all be happy. We'll still aim for that 100% on here. So the most common cellular mechanism used for animal virion entry into host cells is phagocytosis, pinocytosis, endocytosis, hemagglutination, or pore formation. Still, we're aiming for 100% here. And feel free to discuss. You've got another 50 seconds to talk to your neighbors. Yeah, no, yeah, no. 10, 5, 96%. Now, sometimes, actually, I thought this was happening in molecular biology. People were just messing with me, putting down another answer to make sure I couldn't.
People that are negating the point.
Yeah, we could always do it again. That's true. We could. So we didn't talk too much about phagocytosis. This is the much more non-specific kinds of interaction that Ellis was talking about, just cells picking up stuff from the environment. Pinocytosis is also very similar. Again, we didn't talk too much about that. Yes, it is endocytosis. Hemagglutination, that's a great way of detecting viruses. Pore formation, this is actually probably the second best answer here. But that's not necessarily the entry process. So often the pores are going to form, from the endosome into the cytoplasm. So not necessarily the entry getting into the cells. So let me select the answer, close this, because that's the way, at least in theory, it's being maintained. And we can go from there. Yeah?
What do you call the drilling into the bacteria?
Oh, the drilling into the bacteria. What would I call that? Good question. Maybe I should go and look at some of my old exams in terms of what I tried to call that. I would often actually call it a drilling process. So literally this drill, or a lot of people also talk about like a syringe. So a syringe compaction. It's pretty specific for what's happening with bacteriophage. So it's hard to talk about that as being much more of a cellular process. It's not a cellular process.
So is it it's disrupting the cell wall? It's not actually fusing or using any of the cellular membranes as far as getting into the cell wall. It's disrupting it. It's damaging it.
Yeah, so the question here for the people, you guys who didn't hear in the back, but yeah, it is there. They are actually forming a hole in the membrane of the bacterium. And that's clearly also an issue, because if you have holes in your membrane, how is the bacteria actually going to survive? And so what happens in the case, and you can see it actually quite well there with that T4 animation, is the outer membrane is actually mostly intact, whereas the inner membrane has a really obvious hole in it. Yeah?
Is that really much of a problem if you consider the size of the virion compared to the size of the bacteria?
So the question is it really a problem with little holes that are being formed in the outside relative to the bacteria? What often happens in these cases is not just one. If it were just one, it probably wouldn't be such an issue, although certainly leakage from the cell is a big problem. But usually you're going to have many more of these. And so in that case, it does seem to be more of an issue. Yeah?
Was that a conformational, did you say what caused the conformational change?
Oh, so what's happening in terms of what causes the conformational change? It's clearly not a pH change or anything like that. So the conformational change that happens with T4 and, in fact, a lot of the other bacteriophage is just receptor interaction. So the virus protein interacts with the receptor. That causes the conformational change. And in the case of T4, you've got two different ones. You've got the ones, the long tail fibers, and then the short tail fibers in the middle. And so there's first a conformational change after those bind, and then a second conformational change after the middle ones bind. And that then leads to the whole syringe body, basically. So, yeah, they're not so much pulling them together. It's more. These are the tail fibers are like this. They'll compact, and that brings down the internal tail fibers. Those then interact, and that then causes the whole compaction process to take place. Yeah.
Are the syringes like a generic one kind, or is it specific to the cell wall and membrane that you're penetrating?
So the question has to do with, basically, are there differences between these different syringe types relative to the host that they're actually infecting? Definitely for the gram-negatives, those that have those two membranes, it's actually pretty similar with a lot of these viruses. Gram-positives, having a different wall, are actually quite different in terms of how those interact. Yeah?
It looked like the head of the drill dislodged and was in between the two membranes when it first penetrated.
So we'll talk more about that a little bit. Well, maybe more, maybe less. So it turns out that the tip of the drill actually is an enzyme which breaks down the peptidoglycan, which is in between, and helps open a hole in the bottom. So it's compaction and the enzyme part of the drill at the bottom. How the heck do you evolve something like that? Good question. I don't know. Evolution. Yeah. Evolution. It's all about it. So just wanted to talk really briefly about the flip side of entry, exit. Number of different ways. The classic one that everyone looks at, you look at virus replication cycles, is lysis, where the whole cell bursts open and all the virions are released. That's actually probably not the most common way, even though it's the way we always put it in our textbooks, the way that these virions go. Certainly true for a lot of non-enveloped viruses because they don't need to pick up any kinds of membranes. T4, a lot of the bacterial viruses will replicate in this fashion. There is a pretty major problem with this whole lysis process for getting out of the cells. The timing has to be exactly right because the virions depend on, or say the virus really, on all of the cellular metabolism still functioning in order to make enough of these infectious virus particles. But at the same time, making the proteins, or usually proteins, that are involved in having this cell lysis take place. So if you lyse the cell too soon, blast it apart, you're never going to be making any infectious virions. So it's this balance between how fast you're making any of the virions. And in the case of something like T4, this thing's pretty darn complicated. It actually does take quite a long time to put the whole thing together, relatively speaking. So the timing is really important in terms of these lysis. On the other hand, probably much more common are these budding processes, which actually don't kill off your host cell, or they just slow it down in such a way that you're going to continue to be able to produce infectious virions. This is, again, much, much more common probably, even for a few of the bacterial viruses, but definitely for most of the animal viruses. So it's much less critical, all the timing. The issue that you have with budding of these enveloped viruses, and someone asked me this, I think, after a lecture last time, is Are they just budding at random all over the cell? Or are they budding in specific places? And it turns out that they do end up budding in very specific places. And actually, I was reading a paper the other day where they talked about the buddosome. So these zome people, they just get out of control. But it's a specific place in whatever membrane it is, usually the plasma membrane, that the virus is going to be budding through. That's where all the virus glycoproteins are, those that are going to be in the envelope, that are really important for the entry process, of course, for the next round of replication. And we will talk very briefly at the end of the term about these absolutely amazing virus exit mechanisms called virus-associated pyramids. And I'll just leave it at that, and we'll talk about it later. Yeah, Ian, and then I'll get to you, sir.
So you said that it's almost always the plasma membrane. Where else would it be from?
So the question is, what about the exceptions? He always asks about the exceptions. We'll talk about some of these later on. The classic example is herpes viruses. Herpes viruses actually bud through the nuclear membrane, and then they pick up membranes. There are some viruses that we're not ready to talk about too much more detail. We're going to pick up membranes from endoplasmic reticulum, but pretty much any intracellular compartment or the plasma membrane where you're going to get budding. Now, what happens if you get budding from one membrane and how you interact with other membranes? We'll talk about that when we get there. It's really cool.
So for our purposes, budding is most common in animal viruses.
Yes.
And then lysis is most common in, or more common in bacterial viruses?
Yeah, so, okay, the whole, what do I need to know about this for the exam? No, what kind of question would he ask? No, sorry. I want to put you on the spot there, Sarah. But that is, in general, I would say bacterial viruses are going to replicate by lysis. Animal viruses, some will replicate by lysis, some will replicate by this budding process.
What about archaea?
Pardon?
What about archaea?
What about archaea? Great question. We don't know. So some go through these virus-associated pyramids. Some seem to go through lysis. Some seem to go through budding. But generalizations for archaeal viruses, we know so little about archaeal viruses, it's very hard to say. That's why we're working on them.
Yeah.
Or something like that. So, yes?
Just for clarification, when we say animal viruses, are we meaning any eukaryote or specifically animals?
Okay, so the question here is actually a very good one. Animal viruses or eukaryotic viruses in general. 90+% of the eukaryotic viruses that people study, actually probably more like 80%, are animal viruses, 18% are plant viruses, and 2% are the other ones, if that. So, which of course doesn't reflect eukaryotic diversity by any stretch of the imagination. But certainly the animal viruses are replicating this way. Plant viruses, we'll talk about those later. It's a very different story as far as what's going on with plant viruses. And we know practically nothing about viruses that are infecting eukaryotes that are not animals and plants. I think it's a wide open field. I keep trying to recommend this to postdocs, something they should do. There's some really fascinating things that I think can be learned there. The one exception to that is the amoebal viruses, these ginormous, massive things that we will talk about towards the end of the term. And those actually mostly replicate by lysis. But the animal viruses are almost all going to be going through these budding processes. Okay, more questions on entry and then release and stuff. So now we're going to kind of switch gears completely and actually start talking about individual viruses and how they're actually replicating. And the basic process now for the rest of the term will be start small, get big. And probably the smallest of those are these, actually I should have taken out the single-stranded DNA phage here, RNA phage. So it turns out that there are relatively few bacterial viruses that use RNA as their genomes, but those few that have been studied are incredibly well studied and incredibly well known and are really easily applicable to a lot of the RNA viruses that are infecting animals and plants. And so we'll spend the rest of today's lecture and maybe the beginning of Monday's lecture talking about these small RNA phage. And anytime you're talking about RNA viruses, you're always going to have this issue between replication, and when I say replication here, I mean genome replication and translation. So the genome itself gets translated, and you've got to replicate the genome. And how do you deal with these two kind of competing things? And again, it's very well known for some of these RNA bacteriophage, and also quite well known for a lot of the RNA viruses that are infecting eukaryotes. So the key concepts here, again, we'll talk a little bit more about phage. Just got an invitation to the 100th birthday party of phage in Paris. I'm not going to make it. It's in 2 weeks. Anyone flying to Paris? Go in your carry-on bag. So the big deal here with pretty much all of these RNA viruses and the single-stranded pieces, which we've talked about quite a bit already here, you're all very clued in, is a lot about secondary structures. So it's not a single strand. It's all how those single strands are interacting with each other. And then, again, not terribly surprising, there are lots of host proteins that are interacting with these viruses in terms of the virus replication process. And just a quick background here, phage. Phage are bacterial viruses. They eat the bacteria, phagos from the Greek to eat, so literally eat holes in your bacteria. We looked at those plaques, and so some of the very first plaques that were ever seen were the holes that had gotten eaten in the lawn of bacteria by these bacteriophage. So the 100th birthday party that we had 2 years ago was celebrating Twart's discovery of the bacteriophage, and the one that's happening in Paris was the 100th birthday party 2 years later of when Félix Terrel, who was really the one who was the major proponent of not only the study of bacteriophage, but also using bacteriophage for phage therapy, so using the bacterial viruses to treat bacterial virus disease. And this is his major monograph. I think it's only 130 pages in French. Le bactériophage, son rôle dans l'immunité. And so immunity of the host, really, in terms of thinking about the infection, and so using bacteriophage to treat disease. This is not their classic immunity. And again, this was in 1917, so it was a few years back, and immunology has changed a bit since then. Yeah?
Are all of the viruses that infect bacteria called phages?
Yes, so all the viruses which infect bacteria are known as bacteriophage. And most people will contract that to phage. I should really be saying bacteriophage. You could also say bacterial viruses. These are synonyms. Now, don't start calling archaeoviruses phage. I may beat you up. There's a whole long story about that, but we'll maybe get to that when we're done here. So, small RNA phage. When I talk about each of these individual viruses and the virus families that are here, and this is basically the chapters out of our textbook, I'll sort of do this review. Usually it's going to be, how were they found, what does their structure look like, and then some aspects of the molecular replication things, and then sometimes some cool stuff here at the end in terms of the biotechnological applications. So where did they find this particular bacteriophage? Where's a really good place to find bacteriophage? Where do you find a lot of bacteria? Sick people is one place. In your mouth. What comes out of the other end of you? Sewage treatment plants. Sewage treatment plants are absolutely wonderful places to find bacteriophage because they're massive numbers of bacteria, and they're all in these very high concentrations. Yeah, maybe a little bit stinky. If you don't like your students very much and you want to detect a few more bacteriophage, go send them off to find these things. So turns out that there are so many of these small RNA bacteriophage in sewage that people actually use these viruses to detect sewage. So if you're out at the beach, there's some water flowing across the beach, you want to make sure that it's actually clean. One of the ways that people do that, they look for these viruses in there because they're so ridiculously common in sewage. And the way they do that is plaque assays. Very straightforward technique for looking at these. So they can be anywhere from 100 to tens of millions per milliliter in just a standard sewage sample. Not that I want to collect too many standard sewage samples. Yes?
Do you want to make your plaque detection based on E. coli generally? Or?
Yeah, so the question basically is what the host you would use for one of these things.
Plaque.
Yes, and so they are, you're using E. coli for this. And a lot of times people, when you also talk about sewage contamination, people talk about coliform bacteria, which are found in the sewage. And so not surprisingly, it's those coliform bacteria that are infected by these single-stranded bacteriophage. Yeah, so you're exactly right. You're going to use the E. coli as the lawn for your plaque assays. This was where they were found originally. One of the questions that people were trying to answer at the time, this was the very early days of molecular biology. They discovered the fertility factor in E. coli. That's what makes the almost obscene pilus, which goes between various different bacteria and causes then this genetic transfer to take place. Incredibly useful for bacterial genetics. And so people were interested in trying to find these phage that were specific to pilus expressing bacteria, because they thought it'd be really useful in terms of genetic tools moving DNA or RNA around. There are a couple of these that have been well studied. F2 is one of them, and Q beta. Just silly names, but that's where they were found. The Q beta one is slightly larger than F2, and one of the reasons that people got really excited about these, opposed to detecting sewage contamination, was that they turn out to be really nice sources of RNA. You can get massive amounts of really pure defined RNA just by growing a bacterial culture, infecting it with one of these viruses. You end up with a ton of RNA. If you're interested in trying to figure out the genetic code from RNA, you're interested in looking at translation from RNA, this is a really good source of that particular genetic material. So what do these genomes look like? They're tiny, not quite as tiny as some of the viruses and virus genomes we'll talk about later. And this is, again, are they alive? Are they not alive? With just 4 proteins. All you need are 4 proteins to be able to replicate and make the whole virion. The critical ones here, of course, are your coat protein. What's that do? Makes your capsid. Then you need to make more of your genome. Again, these are RNA viruses. There's no RNA-dependent RNA polymerase, at least not ones that are active enough to replicate whole genomes here. Very often, people will call these replicases to differentiate them from the DNA polymerases, the RNA polymerases, which are the standard cellular ones. And basically, all you really need for a virus are these 2 things, your capsid protein and your replicase protein. In the case of these viruses, this is MS2, which is very closely related to F2 and Q beta down here. There are a couple of extra proteins. One of those is a lysis protein. You remember we talked about lysis as being very important for the timing. So you don't want to lyse the cell too soon before you've made all of your different viruses. And then there's a protein called originally called the maturation protein. Which probably has much more to do with entry into the cells. And we'll take a look at that in just a second here. Q beta is slightly different. Doesn't have this specific lysis protein here, but what's called a read-through protein. We'll see this happens quite a lot in various different RNA viruses. There'll be a stop codon that is sometimes used and sometimes not used. That's well known in Q beta. Turns out that this protein actually does a very similar job to this lysis protein. Yeah?
And so MS2 is F2?
MS2 is extremely similar to F2. F2 is the original one that was found, but MS2 is the one that's been much better studied. And it turns out, maybe getting back to, you may be asking a question about the sewage treatment. It's not just F2, it's any of these RNA phage. So there are lots of different ones which are there. So that's what you've got. What do you put it together to make? T equals 3, not quite a completely nice quasi-equivalent icosahedrally symmetric structure, because the maturation protein actually is sitting in this structure. Up at the top here, maybe let's get our pointer out. Go back one here. If I were to give you, or someone were to give you, a question like this in an exam, how would you know that this was T equals 3? You would start up here at a 5-fold axis of symmetry. You'd find another 5-fold axis of symmetry down here, and you try to get from one to the next by hopping in one direction, h of 1, changing directions, k of 1. So it's a T equals 3 regular icosahedron, except for the fact that, again, you've got this maturation protein, and we know that the maturation protein is absolutely critical for getting the virus into the host. And it really is serving basically as the receptor binding protein. So it's got to be on the outside here. And interestingly enough, this particular image up here was made missing the maturation protein. It was just the coat protein, putting all the coat protein together. Everybody remember how many coat proteins you would have in this particular case? T equals 3, assuming that you're missing your maturation protein? 180, exactly. So T number times 60 will give you the number of coat proteins. Now, that's interesting because we got one genome, right? And 180 of the coat protein, one copy of the maturation protein, and actually relatively small numbers of the other proteins as well. So how do you get all these various different amounts of these different proteins? And we'll probably end up mostly talking about that on Monday. But before I forget, this used to be the highest resolution structure of MS2, and we didn't know where the maturation protein was until earlier this year. This is January 2017, where people did cryo-EM structures of single-stranded RNA viruses. In this case, it was actually MS2. And they were able to localize the maturation protein, which is this one, which is here in pink, and see how it interacted with the receptor on the cell, which turns out to be the F-pili. As I mentioned before, this is how they found these viruses in the first place. It was F-dependent bacteriophages. So the purple maturation protein, which is in here, turns out to be in one place. You only have one per virion. Interacts directly with the F-pili structure. The rest of the virion is all T equals 3 pseudoicosahedral. And one of the really amazing things about this structure is they also were able to identify a lot of the RNA present on the inside of each of these capsids. So that's, again, the latest, greatest structure. Definitely far too recent to get into our book as far as that's concerned. So this, well, come on, let's get back to play. So interacts with the pili. We just saw that already. It's through the maturation protein itself. Here, if you look at a poor E. coli infected by one of these viruses, I think this one is MS2, but it could be Q beta, it doesn't really matter. Here are the pili that are coming off of the E. coli, and they're completely encrusted in all of these virions here. So maturation protein is the virus receptor binding protein. Turns out it gets cleaved probably by cellular proteases. How this virion actually gets inside the cell is not completely obvious. Probably when the F-pili contracts, which is what it does when you're pulling two E. coli cells together, it probably just pulls the whole virion inside the cell. So that's the whole entry process. Compaction of the pili which does, and it's actually quite well known that these pili will extend and contract. Contraction of the pilus with these virions attached to it will get that RNA to the inside of the cell. So quick last clicker question of the day. This is a nice review of our structural work that we've talked about before, which is why is the MS2 virion not truly quasi-symmetric? It's got 4 different major capsid proteins, has a cross-linked capsid, maturation protein, RNA genome, 4 proteins, some in overlapping reading frames. No 100% yet. Going for it today.
You're so optimistic.
Hope springs eternal.
I've decided that after I graduate, I'm going to move back to your molecular biology class and just sit in the back and collect random. And I'm not going to register. I'm not going to have registered or anything. So you're going to have, like, 3 weeks towards the end of the term, you'll be like, somebody has a lot of clicker points. And they're not registered yet. So that's you.
10,
Or I'll just sit outside.
5. Yeah. Okay. Do we have 100%? Who are you? I will run you down and let you know. No, there's only one capsid protein here. So yes, it's the maturation protein because it's asymmetric. Have to be symmetric if you had the truly quasi-symmetric process. There are these 4 proteins, some of which are in overlapping reading frames. And this is actually something that I forgot to emphasize, and we'll talk a lot more about it when we talk about the single-stranded DNA viruses. Since the genetic code is a triplet code and normally not overlapping, if you start at a slightly different place that's not in a multiple of 3, you end up encoding a completely different protein. And that's true for MS2. The lysis protein, the open reading frame overlaps with the coat protein, but it has a completely different amino acid sequence. And this is something we'll see quite a bit as we move on through the rest of the course. So we will stop here. Have a good weekend. Hopefully the sun is still shining.